Swayam Prabha. Digital India. Educated India.
Respected chairperson and audience, for next 20 minutes I am going to talk about on the molecular biology of ependymomas and what are the updates in the last couple of years. Ependymomas are the type of gliomas which morphologically and ultrastructurally recapitulate the normal ependymal lining. They comprise about 3 to 5% of all CNS tumors and 6 to 8% of all gliomas. They are more common in children and young adults, and in children, ependymoma is the third most common tumor of the central nervous system, and they comprise about 10 to 12% of all brain tumor in children. WHO proposed the grading of the ependymoma based on the cellularity, pleomorphism, mitosis, and endothelial proliferation. Necrosis is taken into account only when it is a pseudopalisating. Otherwise, even the low-grade ependymoma can show necrosis. These are some of the examples of ependymoma. For example, this is the subependymoma belt-defined tumor in the lateral ventricles, and it is a grade 1. It has very characteristic morphology, groups of tumor cells with clearing microcystic chains in a very separated by the acellular areas. Myxopapillary ependymoma earlier supposed to be or graded as grade 1. They are very classical morphology. These papillary structures with myxoid background in the papillae, and they have a very low MIB labeling index. The myxopapillary ependymomas and subependymoma earlier were graded as grade 1. I will come to this point. Myxopapillary now are graded as grade 2 in the WHO classification. The grade 2 ependymomas are also well-defined tumor in the lateral ventricle or in the fourth ventricle or in the spinal cords. Ependymoma has one the feature which is very characteristic, that they have the pushing margins as compared to the oligodendrogliomas and astrocytomas. This can be highlighted by the GFAP staining. They show these small canals, rosettes, pseudo-rosette around the blood vessels, and when these rosettes are larger, they are known as the ependymal canals. Some of the spinal ependymoma especially, they show a very astrocytic appearance, and one has to be very careful. Some of them may show rosenthal fibers, but the markers can demonstrate the ependymal nature of these cells. Some may show epithelial appearance, some may show astroblastoma-like, or sometimes they are very cystic, mimics the neuroenteric cyst, and some may show very astrocytic appearance. These are some of the variation in the morphology. Some may show clear cell change, some may show giant cell lipomatous change, pigment which may be the lipofuscin or even the melanin, or this osteocartilage and metaplasia is also common in ependymomas. The grade 3 ependymoma, they mimic the glioblastoma both radiologically as well as morphologically. They are very cellular. They may show these endothelial blood vessel proliferation, and the MIB labeling index is very high as compared to grade 1 and grade 2. Now, one of the best marker for the diagnosis on immunohistochemistry is the EMA, and the other is the EBP, immunopositivity. Both the ependymoma shows ring-like or the dot-like positivity, which is very characteristic of ependymoma. Rarely this ring-like positivity or dot-like positivity can be seen in some of the GVM and in meningioma. So one should be very careful. We compared the positivity of EBP50 and EMA, and we found that the EMI is better for the higher grade and the EBP50 is better for the lower grade ependymoma. The other marker which can be helpful in the diagnosis are the ATRX, which is intact as compared to the astrocytic tumor, CD56 positivity, D240 positivity, which also shows dot-like positivity, GFAP. Some ependymoma can show cytokeratin positivity. They are usually negative for the P53 and IDH1. Now, OLIG2 is a relatively good marker for ependymoma. It is very focally positive, or it may be negative in ependymomas as compared to the astrocytic and oligodendroglial tumor. The intramedullary tumor, as I told, is especially in the frozen, when they look like the astrocytic tumors, they can be misdiagnosed. In the frozen section, sometimes they are very cellular, and one may likely to misdiagnose these are the other tumor. Now, most important prognostic factors are the age, site, and extent of the resections. Grade is also important, but one of the problem in the grading is that this prognosis in 2/3 of the cases can be predicted based on the grading, but in 1/3 of the cases there is it cannot be predicted. There's a lot of inherent problem, subjectivity in the grading based on the cellularity, microvascular proliferation, and necrosis. Now, the most important landmark discovery in the ependymoma was when the Taylor et al. They proposed that the ependymoma at different sites, they arise from the radial glial cells and they have different molecular markers, and the prognosis of these tumors depend upon the site and the molecular markers. So in the last WHO in 2016, the ependymoma were WHO suggested that the ependymoma should be diagnosed like the other brain tumor in an integrated fashion. Later diagnosis should be provided based on the morphology, WHO grade, and molecular studies. And WHO suggested that the subependymoma and the myxopapillary ependymoma, which are well circumscribed, they are grade 1, and the ependymoma and the anaplastic ependymoma, they are grade 2 or grade 3. Some of the variant papillary, clear cell, and tenocytic were also included in the last WHO classification. The new addition was the ependymoma which are RELA fusion positive. So these are some of the examples of the ependymoma, which are very epithelial, cellular, clear cells, papillary, and this is a tenocytic variant, more of the spindling, but this is a grade 3, MIB labeling it is very high. So in the last WHO classification, although this classification has come at that time, but it was not totally integrated in the WHO classification. In this classification, based on the methylation profile and some of the epigenetic changes, the ependymoma were grouped into 3 compartments: supratentorial, posterior fossa, and the spinal. And in each compartment there was subependymoma. There is these 2 red one in the supratentorial and the posterior fossa, which were supposed to have worse prognosis. They may be grade 2, but even in the grade 2 they have the worst prognosis. They are the RELA fusion positive, and in the posterior fossa they were the type A ependymoma posterior fossa. Now, based on this classification, it was shown that the subependymoma, they are very well circumscribed. Nothing was changed in the subependymoma group. They are very localized, well circumscribed, very characteristic morphology, very balanced genome. Rarely they can show the 6q deletion in the spinal subependymoma. Now, the myxopapillary ependymoma are also classical location. They are in the spinal cord. They are in the cauda equina. They have the very unstable chromosomal pattern, They may show polyploidy and very characteristic morphology. In the last WHO, they were graded as the grade 1 ependymoma. Now coming to the supratentorial, one of them is subependymoma. The other most important group is the Rella fusion. At that time, the C11ORF95 Rella fusion was described, and this Rella gene, this activates the NF-kappa pathway, and it was the oncogenic mutation. The other was YAB1 fusions. So these are the two important group, but later on this gene Rella fusion, it has been shown that the ORF, which was supposed to be the function was not known, now it is well characterized, and its nomenclature is given to this gene. Now this Rella fusion is especially seen only in the supratentorial, not in the infratentorial or in the spinal compartment, and mostly these are in children. They have worse prognosis. One of the very classical morphological changes is the thin-walled vascular channels, the clearing of the cytoplasm, very high labeling MIB-ADEX if it is grade 3, and the oligo-like appearance, perinuclear halo, and some of these nuclei show the nuclear grooving. This is another case of Rella fusion. This is very thin-walled vascular channel. Now there are surrogate markers for the Rella fusion, which is L1-CAM or the P65 NF-kappa pathway. But the best is fish. One can demonstrate the break apart. One can use the break apart these probes, and one can diagnose these cases as the Rella fusion. Now these surrogate markers are equally good, but fish is the best. So in the supratentorial compartment, when we say this ependymoma, WHO grade 3, earlier people suggested that it is called the anaplastic Rella fusion. And one can give the note that this tumor is likely to behave in worse fashion. Now based on this morphology, it has been shown that the clear cell and the vascular variants, it is not only classical of the Rella fusion. They can have the other morphological variations like the meningioma like the spindle cell variant and so on. So morphology is one, but they can have the other morphological variants also. At that time, this CRX, this gene function of the gene was not known, but now the nomenclature is changed and it has been named as the JEFTA translocation. Now it has shown that the REL is one of the fusion partner of this JEFTA gene. There may be some other partner which can show fusion. Now why these Rella fusion positive ependymomas has a worse prognosis? It is not clearly known, but some of the studies, for example, in our study we have shown that the epithelial to mesenchymal transition is more common in the Rella fusion. The stemness markers, nestin and other vascular endothelial growth factors, they are more common in these Rella fusion positive tumor. That may be the reason that the stemness is, these tumor, they don't respond to chemotherapy. Now coming to the posterior fossa tumor, they are, earlier it was proposed that they are group A and group B. The group A, they occur in the younger age, group B, they occur in the older age. Group A are laterally located, they are more invasive, they show the metastasis, and very characteristic is 1Q gain. Whereas group B are midline, they are less aggressive and their prognosis is better. Now 1Q gain can be shown, it has been shown in the other ependymoma, in the supratentorial, in the posterior fossa, but it is very important only when it is a group A tumor which is in the younger children. Now one of the features is that they are laterally located or the medially located, but sometime, especially in our setup, the tumor patient presents so late that it is very difficult to make out whether it is a medially placed or the laterally placed. So this is one tumor which is medially placed. This is a very cellular. This is endothelial proliferation, mitosis, and they shows very high MIB labeling index. Now the other marker which has been proposed for the classification is S3K27ME3. If it is lost, it is a group A. If it is intact, it is group B. And when there are other surrogate markers, for example, the CXORF, which is overexpressed when ME3 is lost, and the EZH2 is also overexpressed. So one can say this is ependymoma WHO grade 3, posterior fossa, H3K, ME3, loss, and people now suggest this is a pediatric type, PF type, pediatric type, the other is adult type. Now it has been shown that these pediatric type ependymoma and this posterior fossa, they show the overexpression of the hypoxia gene signature, which leads to the less methylation of H3K27 and more of acetylation, and that is the reason that ME3 is lost. So now at the moment this antibody is available. The H3K20 ME3 is very important for the classification of the posterior fossa tumor into A and B, rather than doing the FISH studies for the 1Q gain. Now in the proposed classification which is coming in the WHO, now it has been suggested that to have the uniform policy of diagnosis so that the patient can be put in under some clinical trial, they have proposed that the ependymoma should be diagnosed based on the site and the molecular changes. One of the very important thing is that myxopapillary ependymoma, which was earlier graded grade 1 in the WHO classification, has been now changed to grade 2. The other important change in the WHO is this Arabic system of grading. The Roman system 1, 2, 3, they are replaced by the 1, 2, 3, which are the Arabic. So why the myxopapillary ependymoma is graded as grade 2 now? It has been shown in some larger studies that the chances of CS dissemination and local recurrence is very high in this tumor. Sometimes even they show the distant metastasis. So that is the reason that they don't behave like a grade 1 tumor. So that was the reason that they are now upgraded to grade 2. Why they metastasize, why they look recurrence, may be because of the complete surgical excision is not possible. But the reason for the metastasis in this group tumor is not known completely. The other what some studies have, Ari Perry has suggested that sometime the anaplastic myxopapillary ependymoma, when they shows two of the following features, for example, the mitosis more than 5 or equal to 5 per 10 hyperfield, MIB labeling index more than 10%, microvascular proliferation, and spontaneous necrosis. So if any of the two features are present, this is likely to be called as the anaplastic myxopapillary ependymoma. So in the supratentorial location, the ependymoma should be classified as the C11 ORF 95 or what you call JEFTA fusion positive and YAB1 fusion positive. In the posterior fossa, they are divided in two molecular groups, PFA and PFB, and the H3K27ME3 is the most important for the diagnosis. In the spinal cord tumor, if it is a grade 2 tumor morphologically or grade 3 tumor, one should do the MICN amplification and FISH can be done for this. This is one tumor which we have seen a 32-year male, intramedullary in the D3 to D4 region. It has a more cellular, perivascular pseudorosette, some necrosis, some mitosis, and we did these are some of the markers for the ependymoma. EM is positive, dot-like, EBP is 50 is positive, MIB is very high, and FISH shows the anemic amplification. So this N amplification is not seen in all ependymomas, rare in the spinal cord. But whenever this is amplified, this shows that this is a worse prognosis. So what WHO recommends in the upcoming WHO book, that the supratentorial ependymoma should be classified as C11 ORF or JEPTA fusion positive or YAB1 fusion positive. They should not be graded into grade 2 or grade 3. If both are negative, then one can give the grade for ependymoma in the supratentorial region grade 2 or grade 3. Same is true in the posterior fossa. If H3K27 ME3 is lost, it is a PFA1, PFA. If it is intact, it is PFB. And any tumor which is not showing this loss or gain should be graded as grade 2 or grade 3. Similarly, in the spinal cord, in grade 2 or grade 3 looking tumor, you do anemic amplification, FISH. If it is amplified, don't grade this. If it is not amplified, then one can give the grade 2 or grade 3. The myxopapillary is upgraded to grade 2 and subependymoma is grade 1. Now, why some of the variants are, for example, these tenicete clear cell papillary variant of ependymoma, these terminology are just morphological variants. These are not the true variant of ependymoma. In one study, it has been shown that while doing the methylation study, many of the tumors are either upgraded or downgraded, and many of them belong to some other categories. Now, in the posterior fossa, this is not adopted, but some studies have shown that in the posterior fossa, it is not only one A or one A and B. There are 9 subgroups, but this has been not adopted in the next WHO. Now, some authors suggest that one should this molecular grading. This gives the algorithm for the treatment of these patients, whether the patient should be put on radiotherapy, chemotherapy, or under trials. After the last WHO update, Michael Taylor, wrote an article, Fall of the Optical Wall, Freedom from the Tyranny of the Microscope Improves the Glioma Risk Stratification. And I think in the upcoming WHO, it is totally applicable to the ependymoma. Now, grading is not very important. It is the molecular changes and the site which is more important. So, probably in the next WHO, which is coming very soon, some of the variants are omitted, and anemic is very important for the spinal ependymoma. Rela fusion is very important for the supratentorial, and H3K27 ME3 is important for the posterior fossa tumor. So I am thankful to Professor Chitra Sarkar, my colleague Vaishali Suri, and the neuropathology lab for doing all this work in ependymoma. Thank you.